Good day, everyone. We are from Group 6, and today we will be presenting our design of experiment project, which is study of cryopreservation techniques, cooling rate, and cryoprotectant type. Our presentation will cover the experimental system, design of experiments, implementation using JASP, and quality control. The main aim of this study is to evaluate the effects of cooling rate and cryoprotectant type on post-thaw cell viability during cryopreservation. Cryopreservation is widely used for long-term storage of cells in biotechnology, medicine, and also research. However, cell damage can occur during freezing and thawing. There are two major biological challenges involved, which are ice crystal formation, which can physically damage cell membranes, and also osmotic shock, which affects cell integrity during water movement. Cooling rate plays a crucial role in controlling intracellular ice formation, while cryoprotectant help protect cell membranes and proteins during freezing and thawing. Therefore, studying these two factors together is important to improve cryopreservation outcomes.
Okay, next up we have treatments. So the treatments in this experiment involve variation from the control condition. We varied the cooling rate from slow to fast freezing and also the type of cryoprotectant including DMSO, glycerol, and also sucrose. So the purpose of these treatments are actually to determine whether alternative cryoprotectants or different cooling rates can improve cell survival compared to the standard condition. Next up, we have factors and variables. So this experiment actually consists of two main factors, where the first factor is cooling rate, which is a continuous variable measured in degrees Celsius per minute. So they have with three levels, 1, 5, and 10 degrees per minute. The second factor is cryoprotectant type, which is a categorical variable with three levels, DMSO, glycerol, and also sucrose. So both of these factors are actually manipulated variables, while all the other conditions are kept constant. Next up, we have control environment and constant conditions. These are to ensure that any observed effects are due to only the studied factors, where several conditions were actually held constant. These include cell type and passage number, initial cell density, cryoprotectant concentration, freezing volume and vial type, and also thawing protocol.
To ensure that any observed effects are due only to the studied factors, several conditions were held constant. These include cell type and passage number, initial cell density, cryoprotectant concentration, freezing volume and vial type, and thawing protocol. In addition, the incubation temperature before freezing was maintained at 37 degrees Celsius, thawing was performed in 37-degree water bath, and the samples were stored in liquid nitrogen at minus 196 degrees Celsius. All the experiments were conducted on the same day by the same operator. Because of this, no blocking was required and a completely randomized result was applied. Other than that, the responses. So the primary responses measured in this study post-cell viability expressed as percentage. This is measured using the triband blue exclusion assay with the hemocytometer. We expect that an optimal cooling rate combined with an effective cryoprotectant will result in high cell viability. A secondary response is the cell recovery rate measured after 24 hours of post-thaw incubation.
Next, we have the design choice and justification of DOE. We have selected the 3 by 3 full factorial design of DOE, and it has been executed as a completely randomized design CRD. Why this design is suitable? Because it has one continuous element, which is the cooling rate, and one categorical component, which is the cryoprotectant. As said in the beginning, our goal is to compare rather than to optimize. Thus, response surface methodology is not necessary in this case. The usage of factorial design also allows us to estimate main effects and also to detect any interaction effects between the cooling rate and cryoprotectant. Then we have the replication and randomization. As for replication, we have three biological replicates for each treatment, where it is done to reduce the random biological variation and also to improve the accuracy of the mean response. As for total runs, we have 27 runs by having three cooling rates, three cryoprotectants, and three replicates. And it is done to ensure that every factor of the combination is fairly represented and also to keep the design balanced. As for randomization, JASP is used to randomize the run order to ensure that the uncontrolled variables are also evenly distributed. And lastly, in this DOE, blocking is not applied as this is a single batch experiment and all samples are assumed to be homogeneous. It is appropriate because this experiment is conducted on the same day by the same operator using the same equipment and regions. Thus, blocking would be unnecessary and it would reduce the degrees of freedom. As for model form, two-way ANOVA model is used and this equation describes how the response variable is influenced.
The design was implemented using JASP software under the factorial ANOVA module. Cooling rate and cryoprotectant type were defined as fixed factor, while viability percentage was set as the dependent variable. This slide shows the run matrix, which includes the cooling rate, cryoprotectant type, replicate number, and measured viability. Although the table may be ordered for readability, the actual run order was randomized in JASP. This table shows an example of the two-way factorial ANOVA output generated in JASP. The analysis evaluates the main effect of cryoprotectant type, the main effect of cooling rate, and their interaction effect on cell viability. This output demonstrates that the DOE was successfully implemented and analyzed using appropriate statistical methods.
Lastly, we have the run matrix. The run matrix is the practical execution plan of the DOE. First, we have the randomized run order. The run order was randomized in JASP to prevent the systematic bias. Each row includes the cooling rate, which is one of the two experimental factors, cryoprotectant, which is the second experimental factor, replicates, which allow the estimation of the experimental factor, and lastly, the viability, which is the response variable that represent the post-thaw cell survival.
So for design properties, in summary, our design has two factors, 27 total runs, a full factorial structure with no confounding, and no center points or rotatability as this is not an RSM design. Last but not least, we have risk and quality controls. So for this one, we have several quality control measures that were applied. Randomization was automated in JASP and blinding was not required due to the objective nature of the viability assay. In the event of missing data, repeat freezing would be conducted. All experiments use the same operator, regions, and calibrated equipment to ensure consistency.
In conclusion, this study presents a well-structured experimental system to investigate the effects of cooling rate and also cryoprotectant type on cryopreservation outcomes. The chosen factorial CRD is suitable, statistically reliable, and also effectively implemented using JASP. This design allows reliable comparison of treatments and supports informed decision making in cryopreservation strategies. That's all from us. Thank you.